Okay, cool. Well, hi everyone. This is Heather and Jesse, and this is our lesson 13, Applying Genetics Problems, and this is the last one, so woohoo. For number one, it says it's talking about wanting to use DNA sequencing via the Sanger method to determine the sequence of the DNA molecule, and on the assignment there, it shows that it is 5'. Let me just make sure I scroll up enough here. So it says 5' TAGT CCAA TGC AG, and so it wants to know what the banding pattern would look like on the gel. And so what we do there is we take the original sequence and we come up with a complementary sequence. And what we do here is the complementary sequence is how we determine the banding pattern. So we put that along the left side with 3' at the top and 5' at the bottom. The negative electrode goes at the top and the positive electrode goes at the bottom. And then you just go through and match up. So for DDGTP, you match up where the G's are, and DDATP match up where the A's are. Same thing for the T's and the C's. And that's the banding pattern. So pretty simple there.
Hey, Heather.
Yeah?
I don't know if you can hear it on the video, but there's like sections where I can't hear you.
Oh, really? Maybe I'll say it again just in case. Stop me if it starts breaking up again.
Okay.
Okay. So we have the negative electrode at the top and the positive electrode at the bottom. And we put the complementary sequence of the primer on the left. And then we match up each of the letters here, for example, DDTTP, with what's shown on the right here. And then we fill that in, and that shows the banding pattern. Are we still good on the volume?
Mm-hmm. Yep, you're doing good.
Oh, can you hear me still, Jesse?
You're doing good.
Okay. So for part B, it says, suppose I use E. coli DNA polymerase 1 during the sequencing reaction instead of TAQ polymerase. And that wouldn't work because the E. coli DNA polymerase 1 will be denatured, and so will the DNA. And the Taq polymerase isn't denatured because it isn't used, and so the reaction won't work. For part C, if you accidentally used E. coli DNA helicase in the reaction and added it to the normal DNA sequencing reaction components, the reaction would just proceed as normal because the DNA helicase is to separate or unzip the strands, but that's unnecessary because it's already been done by the heat in the reaction, and so nothing will happen. Then for part D, suppose you added DDATP to one of the 4 test tubes, but you forgot to add the other 3 triphosphates to the other 3 tubes, and everything else is normal. So what happens is that the reaction would fail, and that's because all 4 of the triphosphates are needed to determine the sequencing and banding pattern, and you wouldn't be able to determine the other nucleotide sequences.
So for question 2, part A, it just wants to know during a typical PCR cycle, the synthesis step is going to be done at 72 degrees Celsius, whereas the annealing step is done at 55 degrees Celsius. AMP superscript R is a common selectable marker that is found in many plasmid vectors, while restriction endonuclease recognizes and cleaves the DNA molecules. Part C, during a gel electrophoresis, DNA is placed in an electrical field. The DNA migrates towards the positive electrode during this process. And for part D, the DDATP lacks the 2' OH group and the 3' OH group. E, Transformation is a term that is used to describe the process where bacteria takes up the DNA molecules from its environment. And for part F, during blue and white screening, IPTG is commonly used to induce the expression of the lac operon, while the XGAL factor is cleaved by the beta-galactosidase to turn E. coli cells blue.
Great. So for question number 3, it says you are interested in using PCR to amplify the region of a double-stranded DNA fragment from E. coli. The sequence to be copied is located between the sequences in bold below. So part A, write the single-stranded DNA sequences that you would use for the 2 primers used in the PCR experiment. Each primer should anneal to one of the sequences in bold. And so what I have here is this primer up here from 3' to 5', CAG, TCG, well, TGC, AAT, TGA, GGCT, 5'. And then I have the second one would be CTG, ACT, CAC, TTC, CGA, G. And for part B, a mathematical equation can be used to determine the annealing temperature that should be used for PCR. This is the equation here. And the 2 important things to note about it is that the percent that you get should be expressed as an integer when you calculate it, not a percent or decimal. And then the N is equal to the total number of nucleotides in the primer. So for the first primer, what you do here is you count out how many G's and how many C's are in it. And when we go through and count that up, what we get is 8 primers. And so that would be 8 primers out of 16 is 50%. So you do 81.5 plus 0.41 times 50 minus 675 over 16, the total number of nucleotides in the primer, and you get 59.81 degrees Celsius. For the second primer, you do kind of the same thing. The only difference is that you get 9 out of 16 nucleotides that are either G or C, and when you calculate that out, you get 62.38 degrees Celsius. And then for part C, what are the 2 newly synthesized strands if DDCTP is accidentally included in the PCR experiment? All other appropriate PCR components are found in the reaction mixture, including ATP, DTTP, and DGTP. And so what happens there, so what are the sequences? And so I actually wrote this wrong. It should be TTAAT. And so that one you get by looking at the primer going on the top, going from 3' to 5', and you do the complementary of it, and you stop when you get to the C because what you added was the DTCTP. And so the same thing on the bottom, and then what you do get here is GGG, ATT, AGC, and you stop at the C. Okay, so for part D, so it wants to know why DNAA and DAM are the 2 proteins aren't included in the PCR experiment. And that's because for a couple reasons. So the process requires continuous replication, and DAM would stop that because it methylates it. And so also, I think that I actually meant to write DNAA here, not DAM, helps to initiate the DNA replication, and that's not something that's necessary for what we're doing here. We don't need initiation.
Awesome. For question number 4, it gives you the restriction enzymes, and then it wants you to, for part A, draw an image that represents what your gel would look like after running it. You just look at each restriction enzyme, and on your negative electrode side, you're going to go down from there and start with the biggest fragments. So you'd have 1,000, 500, 400, 300, 200, and 100 base pairs on your side. And then you just put in your restriction enzymes along that gradient, and your larger fragments are going to stay up near your negative electrode, whereas the smaller DNA fragments are going to migrate more towards your positive electrode, so they're going to be farther down on your scale. And so that's what the diagram should look like once you do it for all of your restriction enzymes. For part B, it wants you to draw a linear restriction map to indicate the locations of those restriction enzymes. And so along the map, as you're going along, you're going to have 50 base pairs, and then you're going to hit your BAM-HI enzyme, and then you're going to have 500 base pairs on the other side, and then you're going to hit 50 more base pairs after your other BAM-HI, and then in between, after that, you're going to have your HIND3, HIND3 restriction enzyme, and then 100 base pairs. And then you're going to hit your ECO-RI, and then you're going to have 200 base pairs after that. So you should have, when you're looking at each restriction enzyme, you should add up to the total number that that enzyme has. That would look like. And then for part C, it's really easy. You just look at the different sections that you have for base pairs. And so we had 50, 500, 50, 100, and 200. So that's your answer for part C.
Okay, great. So I have a lot of writing on here, so maybe listen more to what I say than what's on the screen. So number 5, it says you are interested in studying a gene called your favorite gene that you have identified in the yeast Saccharomyces cerevisiae, and it wants you to give the steps for a few different things. So it wants to the steps for generating cDNA for Saccharomyces mature mRNA, clone a cDNA version of YFG into a plasmid vector, and use blue-white screening to identify plasmids that contain the your favorite gene cDNA. So the first thing to generate the cDNA, what you do first is you purify, you need to purify it, and then the next thing you do is you mix the mRNA with the primer. And then after that, you add reverse transcriptase and the DNTPs, and that synthesizes the strand of the DNA. And then the next thing would be to add the RNase H to digest most of the mRNA. And then after that, DNA polymerase from the E. coli synthesizes the second strand of the DNA, and then the restriction enzyme cuts both of them. And let's see here, make sure I'm on the right step. And so, make sure I say this right. Okay. So that was to generate the cDNA. And to clone it, what we want to do is we take the cloning plasmid that was cleaved by the restriction enzyme, and it makes cohesive ends on it. And so the cohesive ends of the DNA fragments that were made up in step 5 up here, the cohesive ends on the DNA fragments and the cohesive ends on the plasmid line up and come together, and then phosphodiester bonds are formed by DNA ligase, and transformation incorporates the plasmids inside into the bacterial host cells, which in this case is the E. coli cells. And the result of that is that separate cells with different segments of DNA from S. cerevisiae are formed, and then that can be put on an agar plate, and you can identify the cDNA version of YFG, and PCR can be used for further replication. And so the other thing is that to use the blue-white screening to identify plasmids that contain the YFG cDNA, what you would do is add IPTG and XGAL, and it will either turn blue or white. And so if it's blue, that means that the LAC-X gene was expressed. And if it's white, that means that the transformation was successful. Okay, that was a lot. Jesse, do you want to add anything to that?
Nope, I think that was it. The only reason why it turns blue is just because you have that intact LAC-Z gene, which is expressing that beta-galactosidase. And when it's white, it's just not intact because it's being disrupted. So that was it.
Okay, great. Well, cool. That was the last one. See ya, everybody.